Welcome from the University of St Andrews to our alumni, colleagues and friends watching from across the globe. I'm Jennifer Head from the Development Office and I work with Science and Medicine faculties at the university. Saints Talks are short presentations by leading members of our academic community on subjects we hope are of general interest. Our Saints Talk today will be given by Professor Maria Dornelas and is titled Nuance in Biodiversity Trends. Professor Dornelas will take questions afterwards. If you would like to ask a question, they can be added to the Teams taskbar. And please don't put your full name for data protection reasons. Maria Dornelas is a professor at the Centre for Biological Diversity of the School of Biology. She became faculty at the University of St Andrews in 2012 as a Marine Alliance for Science and Technology for Scotland lecturer, after postdocs here at the University of Azeredo and at James Cook University, where she also studied for her PhD. She is a macroecologist who researches biodiversity change across time and space. Her work spans millimetre-scale interactions among coral colonies on a reef to time series of biodiversity across the entire planet. When biodiversity makes the news, it's often about loss. Yet looking at how biodiversity has changed through time in the recent past reveals many different paths, with some places gaining and others losing species. Professor Dornelas' talk will explore how biodiversity is changing across scales, places and types of organisms, from the corals at her long-term study site in the Great Barrier Reef to birds, insects and trees across the planet. The talk will explain why there are reasons for hope as well as worry. I read that biodiversity is the most complex feature of our planet and the most vital. So a very important subject. Welcome, Professor Dornelas. We are very much looking forward to hearing your Saints talk.
Thank you very much. Thank you for your kind introduction, and thank you for the people joining us for this talk. So as the title implies, I'm going to talk about the different ways in which biodiversity has changed in the recent past. Right. So when we talk about biodiversity change, we often end up talking about extinction, which is what we call when a species disappears forever and everywhere from this planet. And you may have heard last year, for example, when the first global assessment was published by the IPBES, that an estimated 1 million species are currently at risk of extinction. You may also be aware that current rates of extinction are 10 to 100 times higher than background rates, which would mean that we are in the middle of a mass extinction. The last one is the one that happened at the time when the dinosaurs disappeared. And this one seems to be largely caused by us. But this is not a talk from a prophet of doom. And what I actually hope to show you in this talk is that there are some signs of hope as well as signs of worry. And if we look carefully at biodiversity trends in the recent past, we see like a nuanced mix of gains and losses in different parts of the planet and at different times. We see winners as well as losers among different types of organisms. And what we see is a really complex picture of biodiversity change everywhere. Okay, so I'm going to start this talk by taking you to one of my favorite places on the planet. It's at my long-term study site, Lizard Island in the northern Great Barrier Reef. It's a really beautiful part of the world, and this reef in particular is a reef we call Trimodal, and I've been working there, I've been going there pretty much every November since my PhD. And this reef in particular is actually where I learned to identify corals, which is a tool that I still use today. And more recently, we've started using these torpedo robots, like the one we see at the top there, to build these 3D models of the reef. And they allow us to quantify the structure of the reef and to map out where different species and different shapes of corals live, and this is helping us understand how is it possible that these beautiful ecosystems can harbor so much of the planet's biodiversity. And I want to start the talk by sort of showing you how the reef has changed in the recent past using these 3D maps. And when we started the project in 2014, the island had actually been hit by Cyclone Ita the year before, but this reef had been largely spared, and it was a pretty healthy reef. It had about 40% of its surface was covered by live coral. And in an area the size of a rugby pitch, we could find 110 species, which is a good number. I'm going to point out this like an assuming coral, a massive coral that we see here, because I'm going to use that as our visual anchor as the reef will change through time. So this is 2014. The following year, 2015, the reef was hit by another cyclone, Cyclone Nathan, and it changed profoundly. So most of the hard coral that we saw in the previous image is now gone. Our anchor coral is still there, and there's some small remnants. Most of it is gone, and we see some soft coral over here as well. But it's already quite different from the previous image. The following year, 2016, there was a marine heat wave, and the greatest mass bleaching event on record took place. Bleaching is what happens when the water becomes too hot, and corals rely on microscopic algae for most of their energy. And when the water becomes too hot, that relationship breaks up. The coral expels the algae, it turns pale, hence the name bleaching. And if bleaching is prolonged or very severe, it dies. In this case, most of what was left of the live corals on this reef have died, and you see very little live coral left here. Our anchor coral is still there, though. We can still see it. 2017 saw another bleaching event in this part of the reef. It was not as pronounced as the previous year, but at this particular site, it was the water has actually got hotter. However, there was very little left to die, and so we don't see a great deal of change between 2016 and 2017. But I have to confess that you know, at this point, I felt pretty, I don't know, pretty sad. So this is one of the most extreme sequences of extreme disturbances, I imagine, on record. So we had a sequence of 2 cyclones followed by 2 marine heat waves, which led to over 90% of the corals dying. Losing about 60% of the species that live there, and what is left are really the little smears of coral on the reef. We ended up with less than 2% live coral, and I would really struggle to even call this a coral reef at this point. It's maybe a rocky reef with some corals, but it most certainly does not look like a coral reef to me. And I remember thinking at this point that I'd have to change study sites. I remember feeling very sad at this point in my career because this is a special place for me. I remember not being sure this reef would ever recover because most of the reefs around this reef looked like this. Luckily, I was wrong. In 2018, we went back and we started seeing, you can barely see them here. I used arrows to point them out. We started seeing little smears of coral on the reef, which are baby corals coming back, probably from deeper reefs that we hadn't visited. So suddenly I had some hope, and we kind of changed tact and decided to to study the recovery of these reefs. Our anchor coral is still there. We can still see it. And this is a starfish over here as well. 2019, there was some growth. So some of these smears we can start to recognize as corals that there and there, and there and there. So there's some growth. There's more baby corals coming to the reef. And then in 2020, we can actually start to recognize there's something that looks like a coral reef. So there's, we can see lots of things. We can start to see branching corals and table corals and etc. So there's quite a lot of growth that happened in the past year. There is also a minor bleaching event unfolding at the time of this survey was taken. The most obvious one is this white coral that you can see on the top right corner here, but some of these that are so brighter in color, they're also bleaching. And so I'm actually kind of holding my breath to see what this year's survey is going to look like. I know how many of these guys have survived. It was not as severe as the one we saw in 2016, 2017, but there's definitely bleaching unfolding. At the moment, we can't travel to Australia. Luckily, we couldn't travel last year either. Luckily, we have a team of collaborators based in Australia who are doing the surveys for us so that we can keep this time series growing. And what do we learn from this experience? You know, it's great to be wrong when it's bad news. Two, that time series in biodiversity change are not always in the same direction. We can have some pretty dramatic losses of biodiversity, and then out of nowhere we see all of these small corals coming back, giving us hope that given time, given the right conditions, ecosystems can, actually bounce back from even the most extreme disturbances. But this is one place. It's like, admittedly, like one of my favorite places, but it's just one place. It doesn't tell us what's happening across the world, and that's what I promised on the title of my talk. And in order to deliver on that promise, I actually need to borrow data from lots of different people. So, and that's what we've been doing in the Biotime Database. It's a database of biodiversity time series in the Anthropocene, and it's based here in St Andrews. We've been growing it over the past decade or so, and it collects work of hundreds of biodiversity scientists that are listed, whose names are listed here, and these are the authors of a paper we published a few years ago. So the database is still growing. These numbers are out of date all the time, luckily, because our database managers are hard at work. But it includes over 12 million biodiversity records from over 500,000 locations around the planet, nearly 50,000 species, and it spans from the late 1800s to the present, although most of our data is from the past 50 years or so. And by using this data, we can ask questions about how biodiversity has been changing across the planet more broadly. At some point in the process of accumulating this data, we realized that actually every single one of these nearly 13 million records was an event recorded by a person, a scientist, looking at the natural world and counting a species. And we know from experience from doing field work that often when we're collecting data, we get, we're exposed to all kinds of unpredicted things. So we decided to start collecting the stories with the adventures that scientists who collect biodiversity data get themselves into. This is a blog called Storytime, which is available on this website in case you want to hear about all kinds of crazy things that we do when we're in the field. But back to the data, so this is what it looks like in space. It is, like I said, it's still growing. Some parts of the world we have like Europe and North America, we have lots of data. Other parts of the world, we don't have as much, like Africa and Asia, but it's still growing, and it covers like all realms, so rivers and lakes and oceans and mountains and plains, all kinds of organisms, plants and birds and insects, etc. And so we can take this data and start to ask questions how biodiversity has changed in the recent past. Now, the first time we analyzed this data was a while ago now. The paper came out in 2014, and the database was much smaller. So we had 100 studies, and we now have over 400. And there's lots of different ways in which we could look at it. Biodiversity is a complex concept, but one way we often look at biodiversity is by counting the numbers of species we have in any one place, and in this case, looking at how it changes through time, and that's what I'm showing you here. So we have numbers of species on the vertical axis here, and then we have time on the horizontal axis. Now, when we started collecting this data, we were largely expecting to see biodiversity declines, and much to our surprise, what we actually saw was a balance of gains and losses in the numbers of species that we see in any one place. And actually, on average, and that's what this black line over here is, we can't detect change in either direction. So we don't actually detect change in what we call species richness. We do detect a different type of change though, so we decided to also look at the identities of the species that we see at each place, so whether we see sparrows or whether we see hawks, for example. And we use the measure called Jacquard, which goes from 0 when the 2 time points we're comparing don't have any species in common, to 1 when all of the species were there both times. And what we saw was that pretty much everywhere we looked, that number was going down, which meant that we were replacing the species that we have. We have this really widespread, this ubiquitous change in the composition of the ecosystem. So we're replacing some things with other things. Now, the 2 biodiversity metrics that I've just showed you, they can only change when a species goes extinct or when a new species arrives, what we call a colonization. But this actually only corresponds to 3 and 5% of the species that we have in any one place. The remaining 92% being either species that are always there or species that are there sometimes but not others, but are not, don't belong to the other 2 categories. Also, we sometimes, we don't just care about how many species, we often care about how many birds, like how many sparrows do I actually have in my garden, not just whether I have blue tits versus sparrows. And so in another analysis that we did, we looked at population trends, so changes in abundance, which is positive when we end up with more individuals, or negative when their abundances are going down. And what we found was similarly to what we'd seen with species richness, we see a balance in terms of gains and losses. So there's definitely some species in some places that are becoming less abundant, but these are roughly matched by species and places that are actually increasing, are becoming more abundant than they were in the past. And this really puzzled us because every couple of years there's a very famous report that comes out, the Living Planet report, and its headline number is the Living Planet Index, which tells us that there's been a 60% decline in wildlife since the 1970s, and we're like, how is it possible to reconcile what they find with what we find? So one of the things that we have since done in collaboration with colleagues in Canada was to actually use the data behind the Living Planet Index and reanalyze it. And we found a really interesting result in that the 60% decline that is seen in this index is to some extent a statistical artifact of the way in which the index is calculated, and it's driven by just the 2.4% of the most extreme declines in their database. It turns out, actually, that if you look at the raw data behind it, we also see roughly a balance of winners and losers in those data as well. And we also see interesting patterns in the spatial distribution of where we see winners and where we see losers. So I gave you an example of losers before, so corals are not doing very well in the recent past year. I want to give you an example of a winner now. So I'm originally from Portugal, and when I did my undergraduate degree at Universidade de Lisboa, the Iberian lynx, like a big cat that exists in Portugal and Spain, was pretty much written off as being condemned to extinction. By the time I started my PhD, there were 94 left in the wild. Last year, there were 855. So this is a 9-fold increase in less than 20 years. And although the lynx is not out of the woods by any means, 855 is not a very high number. It is, definitely a success that it has increased in its abundance rather than gone extinct. And we need to be able to celebrate these successes in the same way as we mourn the losses, because these successes actually give us hope that there's things to be done about biodiversity. It's not all bad. There is also good things that are happening. Okay, so I talked about spatial variation, and what I'm going to show you next is another analysis that we did where we decided to map biodiversity change to build an understanding of the geography of biodiversity change. We were interested in asking whether there were some places that were gaining and some places that were losing species on the planet. Again, this is done with the Biotime database that lives here in St. Andrews, although by this time the database was much bigger, as it had about tripled in size. This is also a very different statistical approach than what we did before. We used different models with standardized spatial scale, but I was very reassured to see that actually the overall result we'd seen in the previous analysis still holds, and on average across the planet we can't detect gains or losses, so we see a flat trend, and we see that we have roughly about as many losses as we have gains in species richness. And what I've done in this map is color-coded the marine biomes of the world according to whether they are below the global mean, so the warm colors, or above the global mean, the blue colors. And what I see whenever I show this map is that sort of marine regions towards the tropics, so towards the center of these maps, tend to be the warm colors, and then towards the poles we see mostly blue. So what emerges, and this would be consistent with a signal related to climate change, where we see the tropics becoming inhospitable, and we see species migrating towards the poles, where conditions have become more suitable to species that like warmer temperatures. A big take-home message from that analysis is that the marine tropics, in particular, emerge as being hotspots of biodiversity loss. So bad news for the corals. So I mentioned climate change. The next work that I want to talk about is one that looks directly at whether temperature change explains some of what we've seen here. So we extracted temperature time series that match for the same places and the same times as where we have biodiversity time series. For this analysis, we had to focus on the temperate region because we don't have enough data from the tropics and the polar regions to do this well. Okay, so what we see is that on average, not surprisingly, the temperature has warmed. So the slopes are positive, which means that temperature has warmed. It warmed much faster in some places than others, and actually some of our time series actually detect cooling in this particular place and time. And we see variation in the biodiversity time series at the bottom as well. So some places having gained species, some places having lost species. And what we wanted to know is how much of the variation on the bottom is explained by the variation on the top. And what we found was that the warmest areas that were gaining species with warming. And I'm going to sort of remind you, this is for the temperate region only, so it doesn't include the tropics. And this, again, would be entirely consistent with tropical species moving towards the north or the south, depending on which of the hemispheres we are as the planet warms. And just to give you a concrete example, this is a photograph that one of my collaborators, Josh Madden, took in the Sydney Harbour in 2014. And these blobs that we see on this rocky reef are actually corals. They never used to be there. They are there now because the waters of the Sydney Harbour are warm enough to support corals. However, let me point out, this is not a coral reef, okay? This is not what a coral reef looks like. This is what a coral reef looks like. This is what the Chamorro reef looked like at the end of my PhD. And the reason I want to make this point is just because we have gains in some places and losses in others, they're not necessarily the same, right? It's not necessarily good news, and we need to be very careful in how we interpret this balance of gains and losses. So I'm circling back to this idea of the mass extinction. So I told you at the start of this talk that we have elevated extinction rates, and a mass extinction corresponds to losing 3/4 of the species on a planet. How is it possible that we're losing 3/4 of the species on a planet and yet you don't detect any change? Which of us is wrong? Is it us, or is it the people is estimating extinction rates? Well, actually, none of us are wrong. We're just talking about apples and oranges, and the two findings are can be reconciled if we think about scale, and think about scale in two axes. Actually, we think about the time scale first, and then I'll talk about spatial scale in a second. So I talked about this extinction rates being highly elevated, up to 100 times higher than background rates. But there's many species on the planet, and we did some back of the envelope calculations, and we would need to maintain current extinction rates for 13,800 years to actually lose the estimated 9 million species we have on the planet. I'm not arguing we should try that, right? So every extinction risk is real and it's serious. The point I'm trying to make here is that it's not too late. It's not game over. Every species that goes extinct is dramatic, but we can still stop this mass extinction from unfolding. The other scale that I want to talk about is spatial scale. Extinction happens at the scale of the entire planet, whereas the data that I've showed you so far is at local scales. It turns out there's actually enough species and enough individuals on the planet that change at one scale is not necessarily reflected at different scales. And let me illustrate this with some data. So this is a plot of where we have, again, on the vertical axis, change in biodiversity, which is, it's gains when it's blue and its losses when it's pink. And here on the horizontal axis, we have spatial scale, which goes from like the size of your, a small backyard to entire continents. And what we see is that we see a lot more variation in the rate, in both the direction and the magnitude of biodiversity change at local scales than we do at a scale increases. So change in biodiversity at one scale is almost independent of change at other spatial scales. Okay, and that almost concludes the points I wanted to make, just to, in terms of our, what I would like you to take from this talk is that we see widespread change in biodiversity. We don't see loss everywhere. There's definitely reasons for worry. We shouldn't be complacent, but there's also lots of reasons for hope. And I want to thank all of my collaborators and the funders as well. And that's it from me. Thank you.
Thank you. Thank you for your talk, Professor Dornelas. We'll now move on to questions which our audience can type into the chat bar. I'm going to start with Michael, and he's wondering if you use MSA, mean species abundance, as a metric.
No, I didn't use MSA, largely because MSA can only detect losses and not gains. So it's always relative to the abundance at the start of the time series, and so it has, I think, a statistical problem from that point of view.
Oh, thank you. Our next question is from Dick, and he's wondering, he says, corals have been on Earth for a long time, geologically. So why do you say bad news for corals when you find that tropical species are moving towards higher latitudes? Isn't that potentially good news for tropical species like corals?
So absolutely, I do think that range migrations and species moving towards the poles are a sign of adaptation. I also think that I have definitely been shown that that I had underestimated the ability of corals to bounce back, and I'm very happy to have been wrong there. However, the pace of change that we're seeing with the climate and the pace of change that we're seeing with the rain shifts is mismatched. So it's not clear that the redistribution is happening fast enough to prevent large losses of the super diverse ecosystems we see around coral reefs. So just to give you an example, coral reefs cover, I think it's less than 2% of the oceans, and over 1/3 of marine fish species are found there. So it's a really high concentration of species, and so if we lose the ecosystem, it's a big loss.
Thank you, thank you. Another question that we have, it's wondering how are your findings compatible with an ongoing mass extinction?
Okay, right. So I actually had, that may have preempted the answer to that question with my last slide. So I think it's a question of scale. So both time scale and spatial scale, as I explained towards the end of my talk. So the two findings are entirely compatible, and it means that we should take extinction risk seriously, but use it as a tool to help us identify where we need targeted action, rather than as a reason to give up. Right, so it's not game over yet.
Right. Great. Phoebe's wondering, have you linked species losses and gains with ecosystem functioning or processes or services, and seen any global mean trends or any local trends, like for example on Lizard Island? Do you know the ecological impact of old versus new corals, and were they the same species?
So that's an excellent question, and we are actually actively pursuing that question right now. So it turns out it's not very easy to answer that question at the scale at which we have done for biodiversity, because it means that we need to link that with other sources of information, and there are gaps. There's lots of gaps, but we are working on that right now. At more local scales and at Lizard Island, we have actually linked the changes that we've seen in the structure of the corals, and I guess the shapes of the corals that are gone versus the ones that have stayed, to changes in the fish that live there as well. And we have found that smaller fish in particular, so either because they're small juveniles or because they are just species that are small, are particularly affected by those changes. They tend to be the ones that use the structure the corals build to hide from predators, for example. So we see particular. So I guess this is a complicated answer because it is a complicated process. Like it's not a one-to-one relationship, and there's a lot going on in that space. But I think the question is right on the money. So so what happens when we see these change? What are the consequences for function and services?
That's great, thank you. Do you have any plans to obtain data for the areas of the globe that are currently missing from the Biotime Database?
Absolutely, and every time I give a talk, one of the things that I do is ask for more data. And so, like any contacts that people may have of time series, we're interested. We are actively pursuing both, by engaging with the literature in different languages, as well as contacting people in parts of the world where we know are blind spots for us, in the hope of making this resource better. By the way, though, this part, like what we see, so more representation in largely Europe, North America, and Australia is reflected everywhere. So the weather data has very similar spatial biases, and we can still use it for forecasting the weather, for example.
Oh, thanks. Do you worry your results can be used as an argument against conservation actions?
Yes, I do worry. It's a question that comes up a lot. Ultimately, I think a scientist needs to be true to what the science tells him or her, right? So, and what the data show us is this, and so we need to interpret carefully, and we need to tell the story behind the data carefully. But ultimately, the data says what the data says, and scientific integrity needs to be at the core of what we do.
Thanks. Another question that we've had coming in is asking, what recommendations would you make to policymakers, etc., based on your findings?
Oof. Okay. An important recommendation I think I have is that we need to do better from the point of view of biodiversity monitoring. We have a lot of blind spots. There's a lot of organisms that are important to us about which we know very little. There's been a recent controversy regarding, for example, insects, which has really highlighted the fact that sometimes we talk about biodiversity and we're just talking about birds, for example, but we rely on insects for most of our crops. For example, they provide the pollination services, and we have a pretty poor understanding of how insects have changed. So better monitoring, I think, is an important policy. I guess the importance of looking at change at multiple scales, so both the local context as well as the global context of change in biodiversity is important. I'm reminded of an example of somewhere in the world where two adjacent provinces were treating the same species either as endangered or as invasive because it was changing its range, its distribution in response to climate change. So having both a broader perspective as well as the local understanding of the local context I think are important lessons. I guess, and finally, the single metrics are not enough. Biodiversity is complicated, and we need to understand its complexity.
Absolutely. And here's another complex question. What is the biggest unknowns in biodiversity science? So many. I mean, we don't even know for certain how many species we have on the planet. Like, we estimate around 9 million, but we don't really know for certain. The largest ecosystem on the planet is the deep sea. Like in Biotime, we have maybe 2 time series of the deep sea. We have very little understanding of what is happening in many parts of the world. There's a lot to be done. So it's too, like, I can't pick one. I'm sorry. That's great. So our final comment, actually, it's not a question, but it says not a question, but a wonderful talk, and thank you very much. So we're going to finish there, and I'd just like to say thank you, and thank you, Professor Dornelas, for your time, for sharing your expertise and knowledge with us, and for giving us a highly interesting Saints talk. And thank you also to our alumni, colleagues and friends across the globe for joining us. Our next Saints talk will be given by Professor Will Creswell of the School of Biology on the 21st of September. And you can watch previous talks on our YouTube channel and follow us on social media, Facebook, Twitter, LinkedIn and Instagram. Wishing everyone a lovely day and evening from St Andrews, and thank you once more, Professor Dornelas.